Hi everybody, my name is Faith from Full Potential Learning Academy, and today we are going to learn about the cell cycle. Have you ever wondered how we can grow without running out of skin, or how our bones continue growing? When we're born, we're very small and only weigh a few pounds, but then we're able to grow and expand into teenagers and adults. This happens because of the cell cycle. The cell cycle is responsible for maintaining an appropriate amount of cells within our bodies by splitting one diploid cell into two identical diploid daughter cells. And it's able to do this throughout the whole entire body. So what makes up the cell cycle? The cell cycle is composed of interphase and mitosis. As you can see in this figure, interphase takes up most of the cell cycle. It can take about 18 to 20 hours to complete interphase, while mitosis takes approximately 2 hours to complete. Interphase occurs in between mitotic phases. Interphase is a combination of two words, inter meaning between and phase meaning stage. Interphase represents the portion of the cell cycle between two nuclear divisions, or mitotic cycles. Interphase is the preparation phase before mitosis. Interphase is composed of three subphases, gap 1, S, and gap 2. In G1, the cell accumulates energy and chromosomal building blocks for S phase. During S phase, the cell's genetic material is duplicated. In G2, the cell grows and there is duplication of most cellular contents. Now the cell is ready for mitosis. Mitosis has 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase/cytokinesis. In prophase, pro means before. Prophase is the first phase in mitosis and is important for the remaining phases in the cycle. The cell and its contents condense or disintegrate, the nuclear envelope or the barrier surrounding the nucleus breaks down, and centrosomes begin to move to opposite poles. In prometaphase, the chromosomes continue condensing and are getting ready to align at the metaphase plate. The mitotic spindle develops as spindle microtubules lengthen outward from the centrosome. Each sister chromatid develops a kinetochore in their centromeric region, and kinetochores were attached with the spindle microtubules. Metaphase. Meta means middle, and the chromosomes align at the metaphase plate. You can see a zoomed in version of the metaphase plate to the right. Anaphase. Ana means back. Individual sister chromatids, which are now chromosomes, are pulled to opposite poles or the back of the cell. Telophase and cytokinesis. Telo means end. Chromosomes arrive at opposite sides of the poles and begin to decondense to chromatin. The nuclear envelope reforms and the mitotic spindles break down. Then we have cytokinesis. Cyto meaning cell and kinesis meaning splitting. The cell splits into two daughter cells by forming a cleavage furrow in between both cells. Cells can enter the gap 0 or G0 phase when they're not actively waiting to divide. G0 can be temporary until it's triggered by an external signal to enter G1, or it can be permanent, such as eye cells or nerve cells. Here's a summary of everything that we've learned. 1. Interphase and mitosis make up the cell cycle. 2. There are 3 stages in interphase, G1, S, and G2. 3. There are 5 stages in mitosis, prophase, prometaphase, metaphase, anaphase, and telophase/cytokinesis. 4. If a cell needs to be arrested before mitosis, the cell will remain in what is called the G0 phase. Here are some questions for you. 1. What is the role of S phase in cell division? Why must a cell duplicate its genetic material, or chromosomes, before mitosis? 2. What would happen if cells did not divide themselves? Keep in mind that some cells survive only several days. 3. Why is interphase longer than mitosis? What do you think? And 4. If a mutation was to occur in anaphase, what would be expected to be seen in the resulting daughter cells?